Hi, my name is Alana Fisher, and today's date is January 25th, 2022. Today we'll be looking at genes and hormones and the role they play when it comes to sex determination. So just a general overview, each nucleated cell in the body contains 46 chromosomes or 22 pairs of autosomes and one pair of sex chromosomes. This pair of sex chromosomes will direct the development of internal and external sex organs. So as you can see here, the presence or absence of a Y chromosome will determine the direction of gender development. So a female will inherit two sex chromosomes, one from their mother and one from their father, and a male will inherit an X and a Y chromosome, one from their mother, the X from their mother, and the Y from their father. Interestingly enough, in cases of abnormal distribution of chromosomes, the presence of a Y chromosome means that the embryo will be male even if the zygote has multiple X chromosomes. However, a zygote that only inherits a Y chromosome will die because the larger X chromosome contains essential genes that are missing in the Y. On the other hand, a zygote that only gets one X chromosome will develop into a female, but they'll have Turner syndrome because two X chromosomes are needed for normal female reproductive functioning. Now let's have a look at early embryonic development. So this is the weeks 1 to 6. And during the first 6 weeks of development, embryonic tissues are considered bipotential because they cannot be morphologically identified as male or female. So the bipotential gonad has two layers, the outer cortex and the inner medulla. The bipotential internal genitalia has Wolffian ducts and Müllerian ducts, and then there's also an area for bipotential external genitalia. So I've color coordinated these anatomical areas because each area has an important significance when it comes to the development of either the female or male embryo. So highlighted in pink, the outer cortex and Müllerian ducts will be significant when it comes to female development, and the blue, inner medulla, and Wolffian ducts are important when it comes to male development, but we will look at these in more detail later on. But overall, the direction that these anatomical areas take will depend on the appropriate signal. But what is this signal? This is the SRY gene, or the sex-determining region of the Y chromosome. It's most important for male development, as you can see, because the presence of a SRY gene leads to the development of testes, whereas the absence of the SRY gene leads to the development of ovaries. So this SRY gene will produce a protein called the testes determining factor, or TDF, which binds to DNA and activates additional genes such as the SOX9, WT1, and SF1. And this is ultimately the first steps in the development of the male sexual system. An interesting fact is that testicular development does not require testosterone. However, once the testes have been developed, they can produce testosterone, which will have effects on male development later on. So continuing with male development, the testes were a product of the proteins that were created when those other genes were unlocked. And the testes contain two specialized cells known as the interstitial cells and Sertoli cells. These cells are important because they'll secrete hormones that will direct the rest of male development. So as you can see here, interstitial cells will secrete testosterone and DHT, which is also known as dihydrotestosterone. And each of these hormones has specific functions that help with the development of the male reproductive system. Testosterone is important for the development of the Wolffian duct, as we mentioned in the previous slides. And this Wolffian duct will go on to create the epididymis, vas deferens, and seminal vesicle within the male reproductive system. DHT directs the development of the male external genitalia, so that will lead to the development of the glans and the shaft of the penis and the scrotum. Now let's look at the function of the Sertoli cells. So the function of Sertoli cells is to secrete anti-Müllerian hormone, which eventually causes the degeneration of the Müllerian duct. And if you recall from our earlier slides, the Müllerian duct is very significant in female reproductive development, and therefore is not needed if male development is occurring. Moving on to female development, just by looking at this slide, we can see that the presence of hormones is very important when it comes to developing male reproductive systems. However, when it comes to female development, we don't really need any of these hormones. So recall the gonadal cortex from earlier slides. The absence of the SRY gene means that this gonadal cortex will become ovaries. As we saw in the previous slide, the SRY gene is responsible for male development only. Also recall that in the past slide, testosterone and DHT were very important when it came to the development of male reproductive organ systems. So for female reproduction development, there is an absence of this testosterone and an absence of this DHT. An absence of testosterone will lead to the degeneration of the Wolffian duct, and the Wolffian duct was important for male development, as stated earlier. And the absence of DHT will lead to the development of the external genitalia, which includes the clitoris, the labia minora, the labia majora, the opening of the vagina, and the urethra. Finally, the absence of the anti-Müllerian hormone will lead to the development of the Müllerian duct, which will eventually turn into the fallopian tube, the uterus, and the upper vagina. So, as you can see from this presentation, both genes and hormones play a significant role in the development of female and male reproductive systems.